Hi, my name is Stéphanie Tremblay. I'm from Concordia University. I'm going to present my project on differences in frontal white matter microstructure in coronary artery disease patients. So as you may know, coronary artery disease patients are at a greater risk of developing cognitive decline and dementia later in life. And one of the aspects of brain health that is affected is the white matter. It's been shown that white matter changes contribute to the cognitive impairments seen in these individuals. But the macrostructural changes that are commonly studied, so we're talking about changes in white matter volume such as atrophy or an increased lesion load, these changes don't fully explain the impact of coronary disease on cognition. So we need more sensitive neuroimaging metrics to look at the white matter microstructure to be able to capture perhaps more subtle changes in the white matter. The data collection for this project is currently ongoing at the Montreal Heart Institute, so this is an incomplete data set. So far we have 43 coronary patients and 23 healthy controls. These people are all 50 years old and above. They do not have a history of stroke, neurological, or respiratory disorders, and they don't have any significant cognitive impairment. All these participants completed a cognitive evaluation, which assessed processing speed, executive function, working memory, and episodic memory. They also completed an MRI session, which included diffusion-weighted imaging, or DWI, which is the type of data that I'm focusing on for this project. So I pre-processed the MRI data and fitted the multi-shell DWI to the DTI model and the NAUTI model. And by using these two models, we can gain better insights into the physiological changes that underlie the changes that we see. I then conducted the statistical analysis to look at differences between groups in these white matter metrics using voxel-wise two samples t-test with sex and age as covariates. So what I found is that white matter in the frontal lobe, more specifically in a white matter tract called the forceps minor, was altered in the coronary artery disease patients. More specifically, FA was lower in coronary patients than in controls, and orientation dispersion was higher in patients. So the forceps minor is a white matter tract that connects the left and right frontal lobes, and it plays a crucial role in various cognitive functions, including memory, executive function, and visual motor speed. And it has been shown that microstructural disruption in this area was associated with subjective cognitive decline. Then we investigated the association between the microstructure in this region and cognitive performance on all of the four cognitive domains. And we found significant correlations between FA in this region and the performance in processing speed and executive function. The positive correlation indicates that those with lower FA in this region had worse performance in processing speed and in executive function. In this study, we found that coronary patients had lower fractional anisotropy, or FA, and higher orientation dispersion in the forceps minor, which is a white matter tract in the frontal lobe. This suggests that reduced FA in this region is due to greater fiber dispersion rather than to neurite loss, or alternatively, it may be due to the development of another secondary fiber population in this region. We also found that lower FA in this region was associated with poorer performance in processing speed and in executive function. In conclusion, there were detectable abnormalities in the white matter microstructure of coronary patients. It's important to remember that these patients don't have any neurological disorders, but they are more at risk of developing cognitive impairment and neurological disorders in the future due to their cardiac condition. So studying this early stage of declining cerebral health could allow for earlier detection and earlier intervention, which is likely to be a lot more effective. Our research could also allow to identify targets for intervention so that better prevention strategies and better treatments can be designed and implemented in the future. So I would like to end by thanking everyone that has participated in this project, everyone that has contributed. Of course, thank you to all our funding sources. And please do not hesitate if you have any questions concerning this research. You can contact me at this email or on Twitter. And thank you for your attention.